Cell counting is an important step in many cell-based assays for determining cell number and viability. In general, the goal of counting is to understand how much a sample of an unknown cell concentration should be diluted. The most common laboratory tool for counting cells is the Hemacytometer. The Hemacytometer is a counting tool that was originally created for counting blood cells. At the center of the Hemacytometer are two counting chambers, upon which a laser-etched grid can be found. The grid is made up of 9 major quadrants. The 4 corner quadrants are further divided into 16 smaller quadrants. The central quadrant is divided into 25 of these smaller quadrants, each of which is yet further divided into 16 microquadrants. At the far end of each chamber are sample introduction ports, into which the cell sample to be counted is dispensed. On either side of the counting chambers are the cover slip mounting supports, upon which the quartz cover slip rests, usually about 0.1 millimeters above the counting chamber. Since the area of each large square is 1 millimeter squared, the volume contained by each large square is 0.1 millimeters cubed, or 1/10,000th of a milliliter. Before counting, always take a moment to use ethanol and a Kim wipe to clean and dry away any lint, fingerprints, or watermarks from the cover slip and the counting chamber. Then, carefully add a small amount of water to each of the cover slip mounting supports. The surface tension of the water will hold the cover slip in place. Now, gently triturate the cell suspension, or pipette it up and down, to dislodge any cell clumps. Use a micropipette to aspirate about 10 microliters of the suspension, and then load one of the introduction ports with the sample. The solution will be drawn into the counting chamber by capillary action and should cover the entire grid. While the cells are settling, select the objective. Then load the Hemacytometer onto the stage. Next, view the cells under the microscope. If there are fewer than 5 cells in each of the large quadrants, you may need to spin your sample down again and resuspend the pellet in a smaller volume. If the cells are overlapping each other or are simply too dense to easily distinguish from one another, you may need to dilute your sample in a higher volume of solution. Be sure to keep track of the factor with which you are diluting your cells. You will need it in a later calculation. Now, it's time to count the cells. As we talked about earlier, the counting chamber is covered in a laser-etched grid. The lines of the quadrants make it easier to keep track of the cells you're counting. Choose the size quadrant for counting depending on the density of the cells in your sample. For example, if there are a low number of cells, count all the cells in one of the corner quadrants. For samples with a medium amount of cells, choose one of the 16 smaller quadrants. If there is a very high density of cells, count the cells in one of the 25 central quadrants. No matter which quadrant you choose or the density of the cell sample, count at least 20 to 50 cells per quadrant. Not all of the cells will fall neatly within the quadrants. You can count the cells that touch the top and left lines, but disregard the ones touching the bottom or right ones. To get the most accurate count, use a cell counter to keep track of the cells and take the average of the number of cells in 4 quadrants of the same size. To calculate the number of cells in a 1 milliliter volume, multiply the number of cells counted by 10,000 because, as we mentioned before, each large square on the grid is 1/10,000th of a milliliter. If you counted one of these larger quadrants, simply multiply this number by 1. If you counted one of the smaller squares in one of the corner quadrants, multiply this number by 16. If you counted all of the cells in one of the 25 central quadrants, multiply this number by 25. If you happened to dilute your cell suspension before loading the hemacytometer, you will also need to multiply by the dilution factor. Then, to calculate the number of cells in 1 milliliter of this sample, we would multiply the 20 cells in this central quadrant by 10 to the 4th, and 25 to get a total of 5 times 10 to the 6th cells in 1 milliliter of the sample. Now that we know the total number of cells in 1 milliliter of the sample, we need to multiply this number by the total volume of our solution. So, for example, if we have 5 times 10 to the 6th cells in 1 milliliter, then in 2 milliliters we will have a total of 1 times 10 to the 7th cells. Then, to avoid errors from miscounting, uneven distribution of cells, or pipetting errors, load the other side of the chamber and count your cell sample a second time. Counting the cells under the microscope is also a great time to evaluate the viability of the cells in your sample. Trypan blue, a vital stain, is often mixed with the sample prior to being loaded onto the hemacytometer for this purpose. Live cells exclude this dye because living cells are very selective about what they allow through their membranes. A dead cell, however, does not have an intact membrane, which allows the trypan blue to pass through and stain the cytoplasm. To check the viability of your cell sample, dilute an aliquot of your cell suspension in trypan blue, and then load the trypan blue stained sample just like the regular cell sample. Under the phase contrast, the live cells will appear bright and golden and should be counted. Do not count any of the dull, dead blue cells. Calculate the number of cells as before, this time multiplying the final number by the trypan blue dilution factor. So if our original representative sample had first been diluted in trypan blue, we then would have multiplied our total number of cells by our 1 to 10 trypan blue dilution for a total of 1 times 10 to the 8th cells in our sample. When you're finished counting, remember to clean the cover slip and counting chamber. Now that you're a cell counting pro, let's talk about some of the reasons you might need to know how many cells you have in a particular experiment. After isolating cells from a normal or experimental tissue, it's important to know how many cells you've recovered. Here, the investigators will want to know how many splenocytes, or spleen cells, they have isolated from this mouse spleen. When you're performing an experiment to see how two different cell populations react, it's important to know how many of each cell type you're mixing together, like in this co-culture experiment with B and T cells. You will want to know how many cells you have for many other types of cell-based assays. Here, an ELISpot assay is being set up to determine the number of cells in a sample that will secrete interferon gamma, an inflammatory protein, in response to the human papillomavirus. When performing an experiment in which mRNA needs to be extracted or isolated from your cells of interest, it's important to know how many cells you're starting with in order to acquire a sufficient amount of mRNA for the experiment. The Hemacytometer is an inexpensive and relatively easy-to-use tool for counting cells, but it can be tedious and has the potential for human error. The Scepter handheld automatic counter is, as the name implies, a handheld counting device that has an improved counting accuracy compared to the Hemacytometer and that can discriminate both the size and volume of the cells in a cell sample. The TC10 automated cell counter evaluates both cell number and viability, automatically calculates the total cell number in your sample, and allows the cells to be viewed on its readout screen as well. You've just watched Joe's introduction to cell counting. In this video, we reviewed what a hemacytometer is, how to count cells and determine cell viability, and some different reasons you may need to count cells. Thanks for watching, and remember not to count the blue cells.